Hello everyone, my name is Isaac Hayden, and today I will be discussing aldolase at the metabolic crossroads with a focus on glycolysis, gluconeogenesis, aldolase isoform biology, and the transcription control of the ALDO-B gene. The main idea I want to establish from the beginning is that aldolase is much more than an enzyme that appears in the middle of a metabolic pathway. It provides a useful example of how several levels of biochemistry are integrated. At the chemical level, aldolase catalyze a reversible carbon-carbon bond reaction. At the pathway level, the reaction participates in both glycolysis and gluconeogenesis. At the tissue level, mammals express the major aldolase isoforms like A, B, and C with different expression patterns. And then finally, at the gene regulation level, aldolase B expression is controlled by a combination of transcription factors, which we will be going over. So as we move through the presentation, I will compare glycolysis, gluconeogenesis, then get into aldolase reactions itself. After that, I will compare the aldolase isoforms, explain why aldolase B is metabolically distinctive, and then I'll finish by examining how ALDO-B promoter then integrates with the several transcription factors to produce tissue and context-specific gene expression. And the goal is to connect enzyme chemistry with whole pathway physiology, and then connect both of these concepts with gene regulation. So let's begin with the learn objectives. I want to briefly introduce them for the presentation. So by the end of the presentation, we should first be able to compare glycolysis and gluconeogenesis, focusing on their major purposes, reactions, and how they work in opposite directions to help maintain energy and glucose homeostasis. And next we will examine the reversible aldolase reaction and explain why the same enzyme can participate in both glycolysis and gluconeogenesis. Then with the three mammalian aldolase isoforms like A, B, and C, and then relate their expression patterns to metabolic demands of different tissues. A major focus will be aldolase B, including its unique role in fructose metabolism and why the abnormalities in ALDO-B have important clinical consequences. And then finally, we'll move from metabolism to gene regulation by examining how the transcription factors function, and then we will look specifically at how transcription factors, competition, and cellular context can alter promoter activity. By connecting all of these concepts, the overall goal is to understand how enzyme function, tissue-specific expression, and transcription regulation work together to maintain metabolic homeostasis. With those objectives in mind, let's begin. So here you can see the two different pathways of glycolysis and gluconeogenesis. And to understand why aldolase is important, you first need to place it within the two opposing pathways that are shown here, where we have our glycolysis and then our gluconeogenesis. Glycolysis is the pathway that converts 1,6 carbon glucose molecule into 2,3-carbon pyruvate. So you can see that here. So you have the glucose being converted here. It occurs in the cytosol and is used by essentially all cells. The pathway captures part of the chemical energy stored in glucose as ATP and NADH. For each glucose molecule, glycolysis produces 4 ATP, but it consumes 2 ATP to do this during the investment phase, giving a net yield of only 2 ATP. It also generates 2 NADHs, and this makes glycolysis particularly important when cells need rapid energy production. And so glycolysis contains 3 strongly irreversible regulatory steps. These are catalyzed by hexokinase, or glucokinase, phosphofructokinase 1, and pyruvate kinase. These reactions help give glycolysis a strong overall direction under physiological conditions. On the other hand, gluconeogenesis has almost the opposite physiological purpose. Rather than breaking glucose down, it synthesizes glucose, and especially during times of when your body is fasting and you haven't eaten, or you're exercising, so you're burning all your glucose that you stored, you're burning it faster, so then your body needs to be able to generate glucose so that it can continue functioning. Major gluconeogenesis precursors include lactate, glycerol, and glucogenic amino acids. An important point is that gluconeogenesis is not simply glycolysis running backwards. So that's a key point to remember. They're different processes, so they're not the same thing, one going forward and one going backwards. The reversible reactions can oppose in either direction, but the 3 highly irreversible glycolytic steps must be bypassed. Pyruvate kinase is bypassed by pyruvate carboxylase, and phosphoenolpyruvate carboxylase, or PEPCK and PFK1, is bypassed by fructose 1,6 bisphosphase, and hexokinase, or glucokinase, is passed by glucose 6 phosphatase. And then gluconeogenesis is also energetically expensive, producing 1 glucose from 2 pyruvates, requires 6 high energy phosphate bonds, 4 ATP and 2 GTP, as well as 2 NADHs. So the simplest comparison in this glycolysis is primarily a glucose consuming energy producing pathways, whereas gluconeogenesis is a glucose producing pathway that consumes energy. Yet they share many reversible reactions and intermediates. Aldolase is one of those shared reversible enzymes, which is why it sits as an important metabolic crossroads. Next, we'll go over the investment, cleavage, and payoff of the process. So this slide here, you can see it's breaking down everything here, glycolysis, into 3 conceptual phases: the investment, the cleavage, and then the payoff. So during the investment phase, which will be here, ATP, cells spins ATP to activate the glucose. Glucose is first phosphorylated by glucose 6 phosphatase, and then it is rearranged into fructose 6 phosphate. And so you can kind of see, if you follow along the pathway here, and then the phosphofructokinase 1 then uses another ATP from fructose 1,6 bisphosphate. By this point, 2 ATP molecules have now been invested. So this is why this is called the investment stage. PFK1 is especially important because it catalyzes the connected, the committed step to glycolysis. Once fructose 6 phosphate becomes fructose 1,6 bisphosphate, the carbon skeleton is strongly committed towards the glycolytic breakdown. The next stage is the cleavage, so now we'll be here. And this is where aldolase becomes very central in the process. And so the aldolase will cleave the 6-carbon fructose 1,6 bisphosphate molecule into 2 3-carbon triose phosphates. The G3P and the dihydroxyacetone phosphate, or DHAP. So the DHAP does not continue directly through the payoff reaction. Those phosphates isomerase rapidly convert to DHAP into G3P. So therefore, one molecule of glucose ultimately produces 2 molecules of G3P that enter the payoff phase. So then we'll be coming off here. This doubling is important because every downstream reaction reacts twice per original glucose molecule in the payoff phase, and then oxidation of G3P generates NADH, and substrate level phosphorylation produces ATP. And so altogether, 4 ATP are generated in the payoff phase after the subtracting of the 2 that you had to use in the initial investment earlier. So the glycolysis, again, it yields a net of 2 ATP. The key concept for aldolase is that it performs the carbon splitting step that converts a 6-carbon intermediate into the 2 3-carbon molecules that are used for the energy extraction. It does not directly make ATP, but without aldolase reaction, the pathway could not transition from a preparatory phase to the energy producing phase. So when looking at this pathway, I think of PFK1 as the commitment point and aldolase as the structural turning point. So PFK1 commits the molecules to glycolysis, and aldolase physically divides the carbon skeleton so that the payoff phase can proceed. And so next we'll go on to the bypassing irreversible glycolysis. So all that, if you follow this stepwise here, and I'll explain it. So now we can look at the gluconeogenesis and see why reciprocal regulation is necessary, because several glycolytic reactions are near equilibrium, and so enzymes such as aldolase can catalyze the same chemical reaction in either direction, depending on the substrate, product, and concentrations. The problem is that the 3 irreversible glycolytic reactions cannot simply reverse themselves. Therefore, the gluconeogenesis uses bypass reactions, starting with pyruvate. Pyruvate carboxylase converts pyruvate to an oxaloacetate in the mitochondrion. This reaction requires ATP and is activated by acetyl-CoA. And then the oxaloacetate is then converted towards phosphophenols by PEPCK, and this uses a GTP. This bypasses the pyruvate kinase step. So further up the pathway, fructose 1,6-bisphosphate converts fructose 1,6-bisphosphate to fructose 6-phosphate. This bypasses the PFK1, and then finally the glucose 6-phosphatase converts glucose 6-phosphatate into a free glucose. And then so this is bypassing the hexokinase or the glucokinase reaction. And so on the right side here of the slide, it highlights the reciprocal control. This is physiologically essential because liver should not run glycolysis and gluconeogenesis at maximum rates simultaneously. If it did, ATP would be consumed without accomplishing useful metabolic work in a full cycle. And so hormones help prevent that. In the fed state, insulin generally favors glucose utilization and suppresses the hepatic glucose production. And then on the other side, during fasting, glucogen signaling favoring the gluconeogenesis. And so one important mechanism involves fructose 2,6-bisphosphate. And so lower fructose 2,6-bisphosphate decreases the PFK1 stimulation and relieves the inhibition of fructose 1,6-bisphosphate. And so then this will shift the metabolism towards gluconeogenesis. And then cellular energy status also matters here. So high AMP indicates low energy. Under those conditions, it makes sense to stimulate glycolysis and inhibit energy consuming gluconeogenesis. And so conversely, high acetyl-CoA can then activate pyruvate carboxylase and then signal that carbon can be directed towards glucose production. This is an important connection to aldolase, because that aldolase itself does not require a specific bypass. Its reaction is reversible. The direction of carbon flow is established largely by surrounding pathways, substrate concentrations, energy state, and hormonal regulation. So we'll move on here to the aldolase, the reversible reaction, and then you can see the two pathways here in the reaction. So this slide focuses on the reaction that makes aldolase relevant to both pathways. In glycolysis, aldolase catalyzes cleavage of fructose 1,6-bisphosphate into DHAP and G3P. In gluconeogenesis, the exact same enzyme catalyzes the reverse in the condensation. DHAP and G3P combine to form the fructose 1,6-bisphosphate. This is a classic example of a near equilibrium metabolic reaction. The enzyme does not decide whether the cell is performing glycolysis or gluconeogenesis. Instead, the net direction depends on mass action, meaning the relative concentration of substrates and products, and on the direction imposed by the rest of the pathway. Mammalian aldolases are class 1 aldolases, so mechanically a lysine residue in the active site forms a shift base intermediate with the substrate, and that's temporary covalent linkage, and then it helps to stabilize the reaction intermediates and makes carbon-carbon bond cleavage possible. In the reverse direction, the same catalytic machinery supports carbon-carbon bond formation. That chemistry is very important because breaking a carbon-carbon bond under mild physiological conditions is not trivial. Aldolase lowers the activation energy by stabilizing the intermediates that form the fructose 1,6 bisphosphate, and then is divided into the two 3-carbon molecules. And so you can notice here in the reaction itself does not consume ATP and does not produce ATP. Its significance comes from where its significance comes from the triose phosphate pool that feeds the payoff phase. In gluconeogenesis, it rebuilds a 6-carbon sugar phosphate from 2 3-carbon predecessors. This is why the phase 1 reversible reaction, 2 pathway directions, is so useful. The same enzyme chemistry can be supported either glucose breakdown or glucose synthesis. The physiological meaning changes because the metabolic context changes. So why aldolase B is metabolically distinctive? Although all 3 mammalian aldolase isoforms can participate in the fructose 1,6 bisphosphate reaction, aldolase B has an additional metabolic role that makes it distinctive. On the left here, the aldolase reaction shared with glycolysis and gluconeogenesis, it has fructose 1,6 bisphosphate in a reversible reaction with DHAP and G3P. And on the right, the fructose specific pathway that is particularly important in the liver. And dietary fructose is phosphorylated by the fructokinase to form fructose 1 phosphate. Aldolase B then cleaves fructose 1 phosphate into DHAP, and then those products can either central carbohydrate metabolism. This means aldolase B connects dietary fructose handling with the broader metabolic network. And so the clinical importance of this is because very clear in hereditary fructose intolerance, pathogenic variants in aldo B reduce or they'll eliminate functional aldolase B activity. So fructose can still be phosphorylated to fructose 1 phosphate, but that product cannot be efficiently cleaved as fructose 1 phosphate accumulates. Intercellular inorganic phosphate becomes trapped. ATP availability fails, and the energy metabolism is then disrupted. This can interfere with the gluconeogenesis and the glycolysis processes and contribute to hypoglycemia after fructose exposure. Clinically affected individuals can develop symptoms after consuming fructose, sucrose, or some other sugars. Avoiding those dietary sources is therefore central to management of this disease, because this disease illustrates a broader biochemical principle pathway, and it can become harmful when an upstream reaction continues but a downstream step is then blocked. The problem is not the absence of a product, it's also the accumulation of upstream metabolic and secondary consequences of that accumulation. So aldolase B is metabolically distinctive because it it performs the common aldolase reaction while also serving as a critical enzyme in hepatic fructose metabolism. So now we'll go to the the three mammalian aldolase isoforms. So here we compare the three major mammalian aldolase isoforms A, B, and C, encoded by the aldol A, aldol B, and aldol C. All three share general aldolase fold and core chemistry needed to process fructose 1,6-bisphosphate, but their expression patterns and physiological emphasis are what differs. So aldolase A is broadly expressed and is especially prominent in skeletal muscles and erythrocytes. Those tissues depend heavily on glycolysis. Skeletal muscle may require rapid ATP generation during concentration, contraction. While mature erythrocytes lack the mitochondria and therefore depend on glycolysis for ATP, high aldolase A expression is consistent with that demand for high glycolytic flux. And so aldolase B is enriched in the liver, the kidney cortex, and small intestines. Those organs have specialized roles in nutrient processing and synthetic metabolic homeostasis. So the liver can switch between glucose utilization and glucose production. The kidney cortex contributes to gluconeogenesis, and the small intestines participates in first-pass fructose metabolism. Aldolase B is also able to efficiently process fructose 1-phosphate, linking its tissue distribution to metabolic function. And then aldolase C is enriched in the brain and other neural tissues, with expressions also reported in some additional tissues. Its distribution reflects specialization of carbohydrate metabolism in the nervous system. This is important that it is that the isoforms are not merely different names for identical enzymes located in different organs. Differences in sequence, kinetic properties, protein interactions, and expressions allow tissues to tune aldolase function to their own metabolic demands. So when we discuss enzyme isoforms, we are really discussing a strategy for physiological specialization. So preserve the core reaction, but alter where, when, and under what conditions the enzyme is used. So expression patterns and reflect tissue physiology. This slide, you can see here as we discuss some of the different tissues and areas, organs, where these different aldolases can be found, or high concentrations can be found. So this slide translates the isoform table into the organ level physiology, starting with the muscles. Aldol A predominates because these cells require dependable glycolytic throughput. In erythrocytes, one energy pathway among many, it is the primary source of ATP because mature red blood cells do not contain any mitochondria. And then we move to the liver. Aldol B is where the organ's role as a metabolic regulator. The liver processes nutrients after meals, stores and releases glucose, performs gluconeogenesis during fasting and metabolizing. Like when you're working out, a sustainable amount of dietary fructose is not in our systems because we're burning our energy sources. So that's kind of the function of the liver, or one of the functions of the liver. Aldo B is also expressed in the kidneys. This is significant because the kidney contribute to gluconeogenesis, particularly during prolonged fasting and certain physiological stresses. Therefore, aldolase B is present in another tissue capable of producing glucose. So you have two organs that are capable of producing glucose. In the small intestines, aldolase B contributes to the fructose handling before nutrients fully enter the systemic circulation. This helps explain why Aldo B expression is not exclusively hepatic. In the brain, Aldo C is enriched in our neural tissues and supports the carbohydrate metabolism of neurons and glial cells. The larger lesson is that expression patterns reflects tissue physiology, so that's a key concept. A gene's function cannot be understood only by asking what reaction is present. In catalyzers, we also need to ask where the gene is expressed and what the metabolic priorities of that tissues are. Expression patterns can also change during development and disease. This reminds us that enzyme distribution is regulated rather than prematurely fixed. It is tissue-specific expression and therefore an additional layer of metabolic control beyond enzyme kinases alone. So the Aldo B promoter architecture. Now the presentation shifts from enzyme function to gene regulation. The question becomes, how does liver enriched genes such as Aldo B achieve the appropriate pattern of expression? The promoter contains several regulatory elements positioned upstream of the transcription state. Rather than functioning as a single on and off switch, the region integrates the activity of multiple DNA binding proteins. And so here you can see in the diagram, it shows several of the important elements, and a distal negative element around the upstream region can restrain promoter activity. And so the responsive region provides a site where transcriptional activators can influence expression. And so an NFY associated CCAAT element contributes to promoter function. And so this is closer to the transcription start site, is an important region associated with the HNF1 and HNF3 binding sites. The key concept is combinational regulation. The amount of Aldo B transcription depends on which factors are present, which factors occupy the DNA, whether they cooperate or compete, and what cofactors are available in that particular cell. This is appropriate for a metabolic gene. The liver must continuously adjust gene expression to nutritional state, hormonal signals, developmental state, and cellular context. A promoter containing several regulatory elements can integrate those inputs more perfectly than a promoter controlled by only one factor, the transcription start site. Labeled +1 represents where RNA polymerase begins producing the Aldo B transcript. The upstream elements influence how efficiently the transcriptional machinery is recruited and activated. So the architecture of the promoter provides the physical basis for the regulatory behaviors. We will see in the next slide the our HNF1, HNF3, DPB do not act independently in isolation. Their effects energy from the arrangement of their binding sites and from interactions among factors. So we'll go on to that slide here. So in this slide here, it's illustrating one of the most interesting features of Aldo B regulation, where we have HNF1 and HNF3, which these two here, they can influence an overlapping promoter region. So HNF stands for the hepatocyte nuclear factor. These transcript factors are strongly associated with regulation of genes involved in liver differentiation and metabolism. However, their effects on Aldo B promoters are not identical. HNF1 functions as a strong transactivator in the experimental systems described in the studies used for this presentation. And when HNF1 occupies the relevant promoter region, Aldo B transcription is strongly favored. So HNF3 can recognize an overlapping regulatory sequence because the binding regions overlap. The factors cannot necessarily occupy the same DNA sequence simultaneously. This creates competition for promoter occupancy. The competition matters because HNF3 can restrain HNF1 dependent activation. In other words, the identity of the transcription factor occupying the same changes the output of the promoter. And so what this slide is doing, it is summarizing visually as high transcription when HNF1 is favored and lower transcription when HNF3 occupies the competing site. And it's, this slide should not be interpreted as HNF3 being a universal repressor in every gene or every cellular condition. The important point is that the context of this promoter and those experimental observations, HNF3 can antagonize activation and therefore accept a restraining effect. And so HNF3 can also influence DBP dependent activation, which shows that promoter region involves more than one interaction. This is an excellent example of how gene expression can be controlled by occupancy rather simply by the presence or absence of a transcription factor. And so two factors may both be present in the nucleus, but if they compete for overlapping DNA, their relative concentrations and affinities and interactions can determine which regulatory site dominates. So the overlapping site behaves somewhat like a molecular decision point. And so HNF1 occupancy tends to favor stronger Aldo B transcription, whereas HNF3 occupancy can restrain that output. And so now we'll go to CEBP and DBP, alternative cell specific activation. And so the next regulatory relationship involves CEBP and DBP and demonstrates why cellular context matters. So CEBP refers to the CCAAT enhancer, that's binding protein in the family, which includes transcription factors with important roles in hepatic differentiation and metabolism. DPP is another transcription factor expressed prominently in the liver and associated with regulated gene expression. And so both of them in the region of the ALDO-B promoter, but their apparent effectiveness differs depending on the experimental cell type. In HEPG2, DBP can act as a strong promoter, and then this is a transactivator, and so CEBP is comparatively weaker in that context unless the influence of the distal negative element is removed. In primary hepatocytes, the pattern differs. CEBP can be a strong activator, whereas DBP can show little activity in the classic primary culture experiments summarized here. And so why is it important? Because the DNA sequence of promoter is not the only determination of the transcription. The cellular environment changes the result. Different cells contain different concentrations of transcription factors, cofactors, chromatin-associated proteins, signaling molecules, and other regulatory components. Therefore, the same binding site can produce different outputs in different cell types. This is also a useful caution when interpreting experiments. So, hepatotoma, the derived cell line, is valuable for studying promoter mechanisms, but is not identical to normal primary hepatocytes. And so differences between the molecules and in the models can reveal how strongly gene regulation depends on the native cellular environment. And the central concept is alternative cell-specific activation. CEBP and DPB can both participate in ALDO-B promoter control, but the regulatory weight is not fixed. So neighboring promoters, elements, and cell-specific cofactors help determine this, which factor has the stronger function and effect. And so we'll move on to how for transcription factors coordinate ALDO-B expression. So you can see the different factors here. So this slide brings the four required transcription factors together into an integrated model. First, HNF1 acts as a major activating influence. It binds to the proximal promoter region. Here you can see that in the proximal region, and can strongly increase ALDO-B transcription. And so greater HNF1 occupancy therefore favors an active liver associated expression state. And then so HNF3 provides a competitive influence in the promoter context because its binding site overlaps with the HNF1 associated region. And then HNF3 can compete for occupancy and reduce HNF1 dependent activation. It can also influence DPP dependent activation. And so here third, the CEBP contributes its activation. Its effects becomes especially apparent in primary hepatocytes, emphasizing the importance of native hepatic regulatory environments. And then fourth, the DBP provides another activating input, but its strength is context dependent. It can strongly activate the promoter in the HepG2 cells while showing much weaker activity in primary hepatocytes. in the classic experiments. The central DNA diagram is important because it reminds us that these are not four independent switches. They're acting on a shared promoter. And so the final level of transcription refers DNA sites occupancy, competition among factors, cofactors, availability, and cell types. This is what is meant by the combination control. A gene does not need a separate promoter for every physiological situation. Instead, one promoter can generate different outputs depending on the combination of regulatory proteins that are interacting with it. This also helps explain tissue specificity, liver-enriched transcription factors, and hepatocyte-specific cofactors create a regulatory environment in which ALDO-B expression is favored. Other tissues can lack the same combination of abundance of factors, producing a different transcriptional outcome. Therefore, ALDO-B expression is best understood at the net result of several regulatory inputs rather than the action of the master transcription factor. So here's an integrated model from the nutrient state to ALDO-B expression. So we're connecting the molecular details back to whole body physiology. At the far left here, the nutrients and hormonal state, the metabolic demands placed on the liver, change depending on whether a person has recently eaten, is fasting, or is responding to hormones such as insulin or glucagon. Those physiological conditions influence the activity and availability of transcription factors and cofactors. The effect mechanism can involve signaling pathways, changes in transcription factors, abundance, post-transcriptional regulation, and interactions with the transcriptional machinery. The next step is the promoter occupancy. So you have HNF1, HNF3, CEBP, DPB, and other factors that are interacting within the regulatory elements in the ALDO-B promoter. Competition and at these sites will then determine the transcriptional outputs. Change in transcription then affects ALDO-B, mRNA, and ultimately the abundance of the Aldolase B protein being made. Enzyme abundance helps determine the liver's capacity to process carbohydrate substrates, including fructose. The four conclusions at the bottom here will help you to summarize the biochemical story. And then first, Aldolase catalyzes the reversible reaction, allowing the fructose 1,6 bisphosphate reaction to function in both glycolysis and gluconeogenesis. Second, Aldolase B is metabolically specified because it also processes the fructose 1-phosphate and is enriched in the liver, kidneys, and intestines. And then third, HNF1 generally provides a strong activating influence on the ALDO-B promoter, while HNF3 can restrain activation through competition at an overlapping site. Fourth, CEBP and DBP demonstrate that promoter activation is cell context dependent. The overall principle is that metabolic regulation occurs in multiple levels. So that's a key point. Cells regulate pathways, directions through enzyme activity and substrate availability, but they can also regulate metabolic capacity over a longer time, and by challenging how much of an enzyme is expressed. So in conclusion, Aldolase provides a strong example of biochemical integration. At the reaction level, aldolase catalyzes the reversible interconversion of fructose 1,6 bisphosphate with DHAP and G3P. Because that reaction is reversible, it supports carbon flow and glycolysis as well as gluconeogenesis. At the isoform level, mammals use 3 specialized forms. The aldolase A is strongly associated with high glycolytic demand, especially in muscles and erythrocytes. Aldolase C is enriched in neural tissues, and then aldolase B is enriched in liver and the kidney cortex and small intestines, and has the additional ability to process fructose 1-phosphate. And so you can see all that here. The fructose function gives aldolase B important clinical relevance because loss of a normal aldol B function causes the hereditary fructose intolerance, and demonstrating how disruption of a single metabolic step can affect the phosphate balance, ATP availability, glucose homeostasis, and overall physiology. So finally, at the gene regulation level, ALDO-B expression is controlled. HNF1, HNF3, CEBP, and DBP interact with one another at the promoter site and elements in ways that depend on competition occupancy, cofactors, and cellular contacts. The main takeaway is that enzyme function is shaped by more than the catalytic chemistry. We also have to consider isoform specialization, tissue distribution, pathway contacts, and transcriptional regulation. And so aldolase therefore sits at the true metabolic crossroads. It connects glucose breakdown with glucose synthesis, links carbohydrate metabolism with fructose handling, and demonstrates how gene regulation adapts enzyme abundance to tissue-specific metabolic needs. And here are the references that are used within this presentation. I want to thank you for watching and hope you have a good one.